November 22, variant 1, paper 4, variant 1. The first question, figure 1.1, is a side view of a human skull indicating four types of teeth and the jaw. State the function of the human teeth. The teeth chews the food. طب what is the benefit of chewing the food? Why do we chew the food? Because it is considered physical digestion. What is the difference between physical and chemical digestion? Physical digestion is just breaking it down into smaller pieces. Physical digestion, breaking the molecules into smaller pieces. Okay. State the name of the visible outer layer. The outer layer, it is the enamel, outer layer of the teeth. The white layer that you can see in the mirror is the enamel. Explain the process of tooth decay? That's a straightforward question. What happens in tooth decay? Sugary food gets trapped on the surface of the teeth. Bacteria uses the sugar to make anaerobic respiration, produces lactic acid, and then the acid dissolves the tooth enamel. Okay, mammals can be classified according to the position and the shape of their teeth. It shows the skulls of seven mammals. Skulls of seven mammals. Okay, we have seven mammals, and this is the dichotomous key. How do I solve the dichotomous key? I focus on one picture. Let's say, مثلا, we will focus on A. Focus on A. Okay? And then I will follow my key. I will focus on this picture. Let's follow the key. Uh, two or more different types of teeth. Fewer than two different types of teeth. Do I have two or more different types? Yes, I have this type, I have this type, I have this type. So yes, I have two or more different types. Okay. Go to number 2. Is there a wide gap between the front and the back teeth in both jaws? Or there is no wide gap or no wide gap? Let's check. Yes, there is a wide gap in the middle. Okay. There is a wide gap. Go to number 4. Let's go to number 4. No incisors in the upper jaw. Incisors in both the upper and the lower jaw. Let's check. We have incisors in both the upper and the lower jaw. Okay. Both the upper and the lower. Go to number 5. Incisors on the lower jaw is longer, or incisors on the upper and the lower jaw are similar in size. Let's check again. They are similar in size. They are almost equal. So I will choose this one. Similar in size. This is the name. So I'll put in front of it letter A. And you can complete the dichotomous key in the same way. Look at the next photo. Look at the next picture. This one. Okay. Do I have two or more different types? Yes, I have two or more. Is there a gap? Yes, there is a gap. Is there incisors on the upper and lower? Yes. Okay. And then we will complete the incisor on the lower jaw is longer than the upper. Yes, it is longer. This is a tooth. Can you see this tooth? There's a big tooth here. This is an incisor. It is longer than the upper teeth, and you will put a B in front of this point. The killer whale are mammals. State two internal features you would expect to find in a killer whale that is not in a fish. Internal feature. Internal means inside the organism. So what am I going to choose? Now we know that the fish has single circulation, but the whale, since it is a mammal, it will have double circulation. One of the points is that the mammals have double circulation. Also, mammals have lungs while fish have gills. Since he said internal features, you should talk about features inside the organism. What is the group that includes mammals and fish and amphibians and reptiles and birds? All of these are in the vertebrate group. Question 2. Digestive enzymes catalyzes the breakdown of large insoluble molecules. Enzymes breaks down large insoluble into small soluble molecules. Why is it important? What is the benefit of physical digestion? We said it multiple times. Why do we have to change the molecules into small and soluble? Why is it important that the large insoluble molecules are broken down by chemical digestion, which is enzymes? I will say to change it to small soluble molecules, change it to small soluble molecules to be small enough to diffuse through the membranes for absorption. This is what happens in the intestine. The small molecules passes through the walls to the blood. So they have to be small and soluble in order to be able to diffuse through the small intestine. What is the substance that is the solvent for most molecules? Most of the molecules in our body. What is the solvent for most molecules in our body? It is water. Water is the main solvent inside your body. Okay. This is a nice question. We revised during revising our chapters. The activity of two proteases, A and B, was measured at different pHs. Both enzymes are found in the human alimentary canal. Enzyme A, it is most active in acidic pH. Enzyme B is most active in alkaline pH. He said that these are protease enzymes. These, both of them are protease enzymes. Who are they? Who are they? Of course, enzyme A, which is active in this acidic pH, is the pepsin enzyme in the stomach, while enzyme B is the trypsin enzyme from the pancreatic juice. Describe and explain the roles of the two protease enzymes. What do they do? Both of them breaks proteins into amino acids. A is the pepsin. Its optimum pH is 2. I also use the graph. There is no activity for the pepsin after pH 5. It works in the stomach in the presence of hydrochloric acid. So what about the trypsin? Optimum pH is 10 from the graph. The maximum activity was at pH 10. Before pH 3, there was no activity. It works in the small intestine in alkaline conditions due to the presence of bile and sodium hydrogen carbonate. Remember that the bile juice secreted from the liver and the gallbladder is alkaline. It neutralizes the acidity and provides optimum pH for the pancreatic enzymes. Maltase is a digestive enzyme that acts in the small intestine. State the exact location of maltase. Maltase is secreted from the epithelial cells of the small intestine. Epithelial cells from the epithelium. It digests the maltose sugar into glucose. Maltose to glucose. Okay. Question 3. Question 3. Some apparatus that is used to investigate water loss. We took this apparatus before. This is a jar containing a leafy shoot and it is placed on a balance. What will happen when the leaves lose water? It will lose mass. This is how you can measure your transpiration. So, what is the process by which the leafy shoots lose water? The water vapor goes out of the leaves by transpiration. Before you insert them in the jar, it must be cut underwater. You have this point in your notes. Why do we cut them underwater? Because if you cut them in air, the air bubbles will go inside the xylem and block it. I cut it underwater. Why is this? To prevent air bubbles from blocking the xylem. State the purpose of the oil layer. Why did they put an oil layer on top of the water? The oil layer is always to prevent the water from passing. So here, I do not want the water in the burette to evaporate. This is why I put an oil layer to prevent evaporation of water from the burette. Prevent evaporation of water from the burette. Okay. Using the information, describe one method that can be used to determine how much water is lost. How are you going to measure the rate of transpiration? I will measure the mass lost per unit time. Measure the mass lost per unit time. For example, every 5 minutes you will measure the mass. يبقى record the decrease in mass over time. There is another way that you can record the volume of water used over time. Why is this a way of measurement? Because when the leaf loses water, it will absorb other water from the beaker. So the level of the water in the burette will decrease. يبقى you have two answers, either to measure the mass lost over time or to measure the water used over time. Both are correct. Okay, this is the result. The apparatus shown in figure was used to investigate the effect of temperature on the rate of water loss, effect of temperature on transpiration. This is the rate of water loss. This is the rate of water loss. And this is the temperature. طيب, using the information, calculate how much water would be lost from 1 meter square of leaves in 12 hours if it was at 35 degrees Celsius. Careful, this is a combo math question. At 35 degrees Celsius, here I will go up and then go left. Okay. How much is it? It is 0.28. 0.28 per second. طب I want to know how much water lost in 12 hours. So first of all, you have to change the 12 hours to seconds. 12 hours times 60, it will be changed to minutes, times another 60, it will be changed to seconds, and then times 0.28, which is the number you got from the table. Then you will put the answer from the calculator and the unit, please. يبقى in 12 hours, you need to convert the hours to seconds. So I multiplied by 60 to convert it to minutes, then by 60 to convert it to seconds, and then I multiplied it by the rate of water loss. Using the information, describe and explain the effect of increasing the temperature on the rate of water loss. Describe and explain. Using the information, describe and explain the effect of increasing the temperature on the rate of water loss. Okay, the description is from the graph in front of you. As I can see, as the temperature increases, rate of water loss increases. Okay, it was gradual, and then starting from 37, it became steeper increase after 37. This is the description. What is the explanation بقى? Explain why when you increase the temperature, the transpiration increased. We said before that transpiration is evaporation of water followed by diffusion. When the temperature increases, evaporation of water will increase from the mesophyll cells into the air spaces. This is reason number one. Also, heat energy increases the kinetic energy of the molecule, so there will be faster rate of diffusion of the water vapor gas through stomata. But this is my explanation why the heat increases the evaporation of water. Okay? The apparatus shown can also be used to investigate the effect of changing humidity. I can use the same apparatus, but to check the effect of humidity. Suggest why the mass of water does not change. The mass of water in the apparatus does not change when the leafy shoot is kept at 100% humidity. 100% humidity means that the air around the plant has lots of water vapor. It is full of water vapor. Since it is full of water vapor, there will be no diffusion gradient. The water vapor cannot diffuse. Okay? يبقى no diffusion of water vapor as there is no diffusion gradient, which means that the concentration of water in the leaves and out of the leaf is the same. There's no difference, so there is no diffusion. Okay, Was a tricky question for you to think. Even at extremely low relative humidities, the leafy shoot did not wilt. In this experiment, when you put it in extremely low humidity, it did not wilt. However, in real life, in real life, if you put it in low humidity, it will wilt. But why did this apparatus not wilt? Why here it did not wilt? Supposedly, when there is very low humidity, there will be very high transpiration because they're opposite to each other. Why here it did not wilt? يبقى here it did not wilt because it has continuous supply of water from the burette and the beaker, not like in nature, because it has continuous supply of water. Okay. The investigation on the effect of temperature was done at a relative humidity of 20 degrees Celsius. It was repeated in humidity 80%. Here, the humidity was 20%. I repeated this experiment again with 80% humidity. So when the humidity is higher, transpiration will be lower. Therefore, I will draw same shape of graph, but lower than the original one. He asked you to draw it on the graph. Since there is higher humidity, there will be less transpiration. Higher humidity means less transpiration. Sketch your prediction. This is what we did. Question 4. Chromosomes are made of DNA. Describe the structure of DNA molecule. The DNA molecule, what is it made of? Two strands coiled together. Two strands coiled together to form double helix. Each strand is formed of nucleotides. The nucleotide is made up of sugar, phosphate, nitrogen base, which are A, T, G, and C. And then I tell him how they pair. A pairs with T, G pairs with C using hydrogen bonds. This is the structure of the DNA we have in chapter 4. Okay, that's a repeated question. Repeated question. Outline how antibiotic resistance developed in a population of bacteria. I said this question came multiple times. Every time he asks you how the bacteria became resistant, it is by natural selection. How is this? How did this happen? Mutation in the DNA caused variation in the DNA of the bacteria. Some of them are resistant, some of them are non-resistant. When you take antibiotics, all the non-resistant bacteria are killed, while the resistant bacteria have an advantage. They are resistant, they are not affected. So they survive, reproduce, pass the allele to the offspring. Scientists use differences in antibiotic resistance genes to distinguish between different strains. Suggest why the scientists use the differences in the base sequence to classify the strains of the MRSA rather than using the other methods. That's an easy question. He just used a lot of terms, but he is basically asking you, what is the benefit of using the DNA for classification over the other methods? The other methods is comparing the external appearance and comparing the internal anatomy. Why is the DNA better? Because it is more accurate, it is easier, okay? There is no difference in the appearance of bacteria, so you cannot compare the differences in appearance. Okay. Explain why scientists are concerned that some strains of bacteria have become resistant. What is the problem if the bacteria becomes resistant? Why are we afraid of this? Because if they become resistant, the antibiotics will not kill them. All the antibiotics will not kill them, and then they will spread to all the people and they may cause infection. Resistant bacteria will not be killed by any antibiotic. It cannot be treated, and this will lead to spread of disease. This is why we do not like the resistant bacteria. How the use of antibiotics can be managed to reduce the development. How can you reduce the development of resistant strains? That is what you said earlier. Completing the course of the antibiotic. Do not use antibiotics for long times. Do not use antibiotics except when essential. Okay? Develop new antibiotics. Scientists should develop new antibiotics to kill the bacteria. Suggest why. A suggest question, but it is something related to what you took. Why MRSA is unlikely to be transmitted from mother to her unborn fetus. Placenta. Placenta forms a barrier between the mother and the fetus. This is the function of the placenta. The blood of the baby and the blood of the mother do not mix together to avoid the transmission of bacteria from the mother to the baby. Okay, many species of bacteria do not cause disease. Bacteria are very important in many biological processes. Three natural processes involving bacteria that are important to ecosystems: decomposition, deamination, nitrogen fixation, nitrification, denitrification. All the processes in the nitrogen cycle needed bacteria, but these are not harmful. They are useful processes. Okay, useful processes. Question 5. Fires release carbon dioxide into the atmosphere. State one other natural process that release carbon dioxide. Release, not absorb. So release, it will be respiration. Respiration releases carbon dioxide. Or decomposition. Carbon dioxide is a greenhouse gas. I want another greenhouse gas. The other one was methane. Methane that comes out of the cattle farming and the farms usually, and the decomposition of organic wastes. Data scientists used satellite images to analyze the occurrence of fires globally. Sometimes fires occur in forests. So we use the satellite images to analyze the global fires. During a 14-year period, they tracked all the fires that were larger than 0.21 kilometer and therefore visible from space. The table summarizes the data, categorizing the fires by location. The locations include natural ecosystems and land that is managed by people. These are the natural ecosystems and these are the lands managed by people. Agriculture land, يعني land we planted it. Okay, this is the total number of fires that happened in each place. Average expansion rate, which means that how much the fire gets bigger every day. How the space of the fire, how much it gets bigger on land every day. And this is the estimated duration of the fire. How long did it last? 5.4 days, 4.1 days. Every day it gets bigger by 0.6. Okay, let's see. Using the information, compare the data for the two managed locations with the three natural locations. Compare these numbers with these numbers. Compare the natural with the managed location. What can I see? I can see that the natural had more number of fires, more total number of fires. Also, bigger expansion rate. Also, they lasted for longer time. This is the comparison. This is the comparison. The number of fires in the natural ecosystem is higher. This is one point. They have lower expansion. The managed lands have lower expansion rate. The managed lands have shorter duration, or you can write the opposite. Describe how the data could be used to estimate the total area that was burned during the 14-year period for each location. If I want to know the total area burnt, what am I going to do? I will multiply all the numbers. I will multiply the number of fires by expansion rate by duration. Why is this? This is the number of fires, and this is how big it becomes every day, and this is the average duration for each fire. So if you want to calculate how big is the total area burnt, you will multiply all the numbers together. Burning large areas of forest is a cause of habitat destruction. What are the consequences of habitat destruction? What happens when you destroy the habitat? When you destroy the habitat, extinction of animals. Animals becomes endangered. They become extinct. Loss of biodiversity. Disruption of the food chains. Negative effects on the nitrogen cycle and the water cycle and the carbon cycle. All the cycles will be affected. The last question in this exam, part of the human gas exchange system. Okay. And then he asked me, what is the function of the hairs in the nose? It traps dust and particles before they reach the lungs. Traps particles before they reach lungs. Okay. The second question was, what prevents the collapse of the airways? I do not want the airway to close. The cartilage prevents collapse. Cartilage around the trachea and around the bronchus. You can either write J or B. J or B, it was also accepted because there is cartilage around the bronchus. Contracts to decrease the pressure in the thorax. Decrease the pressure. So inside you have low pressure, outside you have high pressure. This means that you are inhaling. Type. What contracts to decrease the pressure? And he gave you the letter F. What is F? F is the external intercostal muscle. The muscle on the outside is the external intercostal muscle. Okay, and then he gave you the word diaphragm. Where is the diaphragm? It is the muscle below the lung, which is C. Muscle below the lung, which is C. What does it do? The same. It contracts to increase the volume or reduce the pressure. Contracts to reduce pressure in lungs. Same as the external intercostal muscle. Okay. What protects the lungs from mechanical damage? Of course, it will be the ribs. Where is the label of the ribs? It is E. Why do I see it as a circle? Because I cut in it so I can see the cross section. This is the cross section of the rib. It will be rib cage or ribs and the letter E. Ribs, E. Contain cilia to move the mucus. I can write the trachea or the bronchus. So trachea, which is J, trachea, J, or if you write bronchus, it will be B. Site of gas exchange, this is the alveoli. Where is the alveoli? These are the bubbles at the end of the bronchioles, which is H. H. Okay. How are the alveoli adapted? I will give him the adaptation and its reason. For example, thin walls for less diffusion distance, large surface area for faster diffusion, good blood supply and good ventilation to have enough oxygen to deliver to the tissues. Explain the differences in the composition between the inspired and the expired air. When you explain the difference, you do not just write the difference. يعني you do not just say inspired air has more oxygen. Okay, yes, it does have more oxygen. Why? The expired has less oxygen because it is used in respiration. The expired air has more carbon dioxide because the carbon dioxide is a product of respiration, waste product of respiration. Okay, so you have to use you have to tell him the reason, explain the differences between the inspired and the expired air. Physical activity changes the concentration of carbon dioxide in the body. Where is this change detected? And how does the body respond to it? When you do exercise, respiration releases lots of carbon dioxide. Which organ detects the change in the carbon dioxide? When you exercise, carbon dioxide increases. This increases the acidity of the blood, which is sensed by the brain, and then it responds by sending impulses to the heart and the lung. So you will increase the breathing rate, increase the heart rate to get rid of the carbon dioxide. The last question was state the name of the solution that can be used to test for the presence of carbon dioxide. You can either say lime water or hydrogen carbonate indicate. And this exam is finished.